Hello and welcome to this presentation in which we discuss the properties of epigenetic information and its associations to mammalian aging. We will be discussing how different systems and treatments enable an organism to age relatively faster as opposed to an organism within the same species. Furthermore, we will be addressing how double-stranded DNA breaks erode the epigenetic landscape and what the reversibility of epigenetic reprogramming can tell us. Okay, so we first begin our discussion with a brief description of how yeast studies have contributed to the epigenetic relevance in aging, as well as some of the phenotypic changes in mice that were observed undergoing the treatment via the eye system. Throughout the 1950s, there was a relationship established between the loss of genetic information and its associations with aging. Independently, Szilard and Midwar proposed mutations from DNA damage that were responsible for the changes in aging. However, this association has been brought to question since some strains of mice or people with higher mutation rates show little to no evidence of premature aging. In the 90s, yeast studies revealed a relationship between the epigenetic information and aging in which DNA stranded breaks, or DSB, were the DNA damage that were most consistently linked to aging. The present-day consensus is that epigenetic changes linked to aging, including the changes in DNA methylation, that patterns are observed in multicellular organisms. The why for the changes of mammalian epigenome over time are still not known, however. So the eye system is a system that generates specific DNA breaks with no evidence of mutations or immediate deleterious effects. Some of the evidence for epigenetic aging with the eye treatment include post-treated eye cells were more susceptible to DNA-damaging agents as opposed to the Cree controls, and hypoinduction and epigenetic age advancement were observable over time as opposed to the Cree control. The results are that the post-treated eye cells were more epigenetically older than the untreated cells. But most importantly, however, were that the DNA breaks were not a result of mutations. Now that it's been established that the eye system is a system that induces epigenetic changes, comparisons between the Cree control and ice mice treatments can be observed. Some of the phenotypic changes observed in the ice mice treatment were alopecia and loss of pigmentation in feet, ears, and nose after 1 month, higher frailty index, including loss in body weight, co-condition, reduced vision, and hearing. For brain changes, some of the observed phenotypic changes include reduced hippocampal function, decline in overall central nervous system function. Within muscle aging, some of the phenotypic changes observed were less muscle mass, reduced endurance, greater lactate levels post-exercise, reduced ATP, and mitochondrial DNA. What can be concluded with these phenotypic changes is that the epigenetic clock serves as the biomarker of biological age in mammals.
I will talk about cellular response to double-strand DNA breaks throughout the epigenetic landscape. First, I will talk about what is the epigenetic landscape. It illustrates the various development pathway a cell might take toward differentiation. The epigenetic landscape integrates the connected concept of competition, induction, and the regulative ability of the genes into a single model designed to explain cellular differentiation. And also, the cell identity is specified by transcriptional networks and chromatin structures. In this study, we find that the loss of epigenetic landscape will cause aging problem. And the loss of the epigenetic landscape includes the DNA methylation and also some genes change. Due to the epigenetic landscape change, it will decrease the H3K27ME3. And also from HOX1 to HOX6, the level of H3K27AC and H3K56AC will decrease, but from HOX9 to HOX13, it will increase. The change for the H3K27AC will cause a change for the stress response, DNA repair, chromatin structure, and metabolism.
I will now discuss how manipulating the epigenome can allow us to either induce aging forward or backward. According to earlier studies, aging in eukaryotes is influenced by the loss of transcriptional network and epigenetic information over time due to certain DNA damage like double-stranded breaks. We all know that as we get older, many of the important biological processes that are vital for survival get altered in a way that leads to a decrease in tissue and organ function, as well as many other factors as a result of aging. One reason that this occurs has to do with the fact that many of the cells of a mature person are usually in a dormant or senescent state as a result of cells no longer capable of restoring function and ability to divide. In normal dividing cells, they possess the ability to detect certain types of DNA damage like double-stranded breaks and repair such errors by entering a senescent state, which will provide cells with enough time to correct any mistakes. Aging is correlated with a deficiency in DNA repair mechanism, and thus, since cells no longer have ways to restore function, DNA damage accumulates, which leads to the loss of epigenetic information. With that in mind, a decrease in lamin B1 has shown to be an important hallmark of aging, which is an intermediate filament protein that provides essential structural support to the nucleus of cells. Such loss of protein results in the initiation of cellular senescence, as indicated by the increase in senescence associated B galactosidase activity found in figure F and G. Now, what is interesting is that ice treated cells showed a decrease in lamin B1 levels, indicating an increase in cellular senescence. In addition to using this technique of accelerating non-mutagenic damages, additional hallmarks or biomarkers have been observed which included an almost similar pattern of gene expression and DNA methylation among ICE-treated mice and untreated older mice than to the younger mice. As shown within the figure, the result of the double-stranded breaks was the loss of important signaling molecules that corresponded with having or turning specific genes on. The initiation of DNA methylation turned these genes off. Here we see that ice-treated mice showed similar altered genes depicted in red and blue as those found in 24-month-old mice than in Cree control mice. With all this evidence, scientists have demonstrated with ice technique that mammalian aging can be driven forward by successfully manipulating the epigenome to create errors and loss of epigenetic information as those found in older mice. Now, as more was understood about the effects of methylation and double-stranded breaks has on the epigenome, scientists wanted to see if it was possible to reverse such effects. In a particular example comparing ice-treated mice with Cree control, it was observed that with aging, the eye lens become more non-transparent as a result of retinal ganglion cells lost, which are essential for propagating important visual cues or information to the brain, and without them, vision became very blurred. However, when this technique of demethylating eye cells came into play, many of the gene expression patterns returned to a more youthful characteristic, resulting in the important activation of certain genes controlling the function and availability of retinal ganglion cells' myelinating axons for the proper conduction of electrical signals to propagate towards the brain. Such effects showed not only that aging can be driven backward, but it provided further evidence that it was epigenetic alterations that influenced aging and not as a result of mutations.